Hello and welcome to this video presentation. My name is Jon Magerøy. I'm a researcher at the Norwegian Institute of Nature Research, and I specialize in freshwater mussels and especially the freshwater pearl mussel. I'm going to give you a talk on the restoration of the freshwater pearl mussel, Margaritifera margaritifera in Norway. I'll start my talk by giving you an overview of the talk. I'll briefly talk about the life cycle of the freshwater pearl mussel, the status of the mussel in Norway. I'll give you an overview of the restoration methods that have been tested, and then I'll go into more detail on relocation of mussels, of fish with mussel larvae, cultivation and stocking of juvenile mussels as restoration efforts. Now, most of us think of a mussel or shell as the adult mussel you see at the top of this diagram. And pearl mussels live at the bottom of rivers and streams. The adult mussels can actually filter 40 liters of water a day, so they're really important for maintaining good water quality in our streams and rivers. But they have quite an interesting life cycle. Female mussels release larvae, and you can see one of those larvae to the right in the diagram. The larvae attach the gills of trout or salmon. They stay on there for about 9 months. Then they metamorphose into juvenile mussels. You can see one of those on the left of the diagram. The juvenile mussels drop to the river bottom and bury into the substrate, where they stay for several years before emerging as an adult mussel. Now, the pearl mussel is found throughout most of the lower elevations in Norway, as you can see from the map to the right. And the blue squares indicate areas where there are living populations of the mussel, and the red squares indicate areas where there's only historic records of mussel populations. We know of more than 600 historic sites, and we currently have 425 sites with live mussels. But there's only sufficient recruitment at 50% of the current sites to maintain the mussel populations, so we are in danger of losing another half of the current sites. Now, we have two varieties of the mussel in Norway, which you do not have in the Czech Republic. We have both salmon mussels and trout mussels, while you only have trout mussels. And the difference between these two variants is that salmon mussels, they primarily infect salmon with their larvae, and trout mussels only infect trout with their larvae. And these two variants are genetically different. The species is red listed as vulnerable due to the loss of sites and the potential loss of half the current sites. I'll give you an overview of the restoration methods that have been tested or used in Norway. We've tried stocking of host fish, both for salmon mussels and trout mussels. This has had the best effect in areas where salmon has disappeared, primarily due to migration barriers being built, such as dams, preventing the salmon from accessing upstream reaches of rivers. But also in some cases where parasites have killed off the salmon, and you've reintroduced salmon, and that has led to recruitment of juvenile mussels. We've also done infection of fish with mussel larvae, and I'll go into more detail on that. We've done removal of migration barriers, and again, that's also been tied to allowing salmon back into their natural areas, so that salmon mussels actually have available hosts for their larvae. We've done relocations of mussels, and I'll get more into detail on that as well. We've also done biotope improvement. We've tried adding rocks, adding gravel to create the correct substrate for the mussels. However, this has primarily been focused actually on the host fish. And it has been shown that it can have a positive effect on the production of host fish, but there's actually been little evaluation of whether this infects the mussel. We've also tried sort of a recruitment channel, a juvenile mussel channel, according to a Czech method. in one of our mussel streams. However, it didn't work that well. The mussels didn't do that well in the channel, and then there was a big pollution event that killed off all the mussels. We also tried livestock impact mitigation. Livestock can crush mussels when they walk in the river, or they can cause siltation, which is negative for the mussels. And therefore, you might want to fence off the river, and this has been tried a few places, but it hasn't really been evaluated. And buffer zones are also sometimes created to reduce the runoff and the siltation in rivers, but it also has not been evaluated, the effect on the mussels. Now, the biggest restoration program that's run in Norway is the cultivation and stocking of juvenile mussels, and I'll get more into detail on that later on. We've also worked some on environmental design with respect to hydropower. We've increased water flows, minimum water flows. We've tried to do flushing floods where you release pulses of water that reduces the siltation in the rivers. There has been very limited evaluation of this, but there has been some cases of where increasing minimum water flow has led to new recruitment of mussels. And then we've also tried to lime acidified rivers. In Norway, especially in the southern part, we've had big problems with acid rain causing acidification of rivers, and it is still a problem. And to combat this, we've been liming rivers. And all of these rivers, we actually thought that the mussel had gone extinct. But then it has been refound, so there must have been some old mussels somewhere that survived, and there's actually been recruitment in several of these rivers that have been limed. Now I'll go more into detail on the relocation of mussels. We've basically done two types of relocations. We've done relocations between rivers, and I only know of 3 relocations between rivers that have been done as restoration efforts. And in one of those cases, the relocation actually resulted in recruitment in the river that used to have a mussel population. Adult mussels were moved there from another river, and eventually you had recruitment in that river. We also had other relocations that were not restoration efforts, and we know of at least 18 rivers where mussels have been introduced. And a lot of them are historic rivers that used to have mussels, and they've been reintroduced. And this has been done for various reasons. In one case, it was done as a wedding gift. They took rivers from another mussels from another river and put them in the river as a wedding gift to the couple getting married. And it actually led to a new population being established in that river. Unfortunately, the old mussel population that was thought to extinct was not extinct. So now you actually have 2 populations in that river. You have the local population and the introduced population. And as some of you may know, I mean, pearl mussels were an important revenue as they produced pearls, especially until the cultured pearl started being produced. So that might have been a motivation for introducing pearl mussels into rivers. If you're a local farmer, you see, oh, they have pearl mussels in that river, they produce pearls, they make money off of that. I might move some mussels to my river and see if I can make some money off of that. However, we don't know how many places this has been done. We have records for 18 places that they've been moved, but it might have been done for a lot more rivers. And we do have one genetic study that showed that 2 mussel populations close to each other, actually in different river systems, were actually genetically identical, suggesting that one of them had been introduced from the other one, but we have no historical records of that. The other type of relocation is relocations within a river, and this can be done as a permanent relocation. That's been done for 14 rivers, and this is often done as a restoration effort, and I'll show that briefly. But it can also be done as a temporary effort. For example, if you're having a road being built, they might have to do some work in the river, so the mussels have been removed to an upstream site while the work has been done, and then moved back after the work has been done. And I write at least 3 rivers here because this is often done by consulting companies, and their notes or reports aren't necessarily made public. So we think that this has been done at a lot more sites that we don't know of. The Leira River is a river just north of Oslo in Norway. Data from 1998 to 2009 show that there's about 25 to 50,000 live eastern pearl shells, and there's very low recruitment in the river. And in the map, the blue line indicates the distribution area of the mussel within the river, and the relocation effort that I'm going to describe to you the most reaches of the river. In 2017 to 2019, almost 6,000 mussels were relocated from the area that's shown in the red circle in the map and in the lower photograph. It's a very slow flowing area, and those mussels were moved up to the area in the green circle, which is faster flowing. And the thought behind this was that there's going to be more fish in the faster flowing area, which provide more hosts for the mussel larvae, and also the habitat for juvenile mussels were going to be better in the faster flowing area. And therefore, this should result in increased recruitment of mussels in the river. The results of the relocation is that, unfortunately, in one of the channels that they moved the mussels to, they had high mortality, probably due to drought. They also tried to look at the prevalence of the mussel larvae on fish gills before and after the relocation. As you see from the data, it's not very different, but further evaluation needs to be done. And long term, one needs to look at whether recruitment actually has increased. We'll move on to looking at the infection of fish with mussel larvae. Here we've also tried 2 different types. There's been infections that have been done within the river. You take mussels, you collect fish from the river, and you put them in a cage so they can't escape. And then the fish releases its larvae and infect, or the mussels release their larvae and infect the fish. And we've done, or this has been done in 10 rivers. And usually the evaluation process has been to see if there's been successful infection of the fish, which normally happens, and then if they can recapture infected fish to look at survival of the infected fish. And unfortunately, the recapture of infected fish has often been very low, and nobody has really looked at recruitment resulting from these efforts. So we don't really know if this method works. Further evaluation of that is needed. Now, you can also stock infected fish from a fish breeding facility, where you take mussels into the facility and you infect the fish. Now, this allows you to stock a lot more infected fish than the river trials. And this has been done 2 places in Norway. For the one place, there has been no evaluation of the effects. For the other place, there has, and I'll get back to that. Now, we've also had unintentional stocking of infected fish. In the Oslo area, there's a local fish administration. They have been stocking trout throughout Oslo and surrounding areas for many years. And until 1994, they didn't have a functional filter on the intake water for their fish breeding facility. And there were many instances of the fish being infected with mussel larvae, because there's quite a big mussel population in the river where they take their water from. So we know that they had infected fish in their facility, and we know that they stocked that fish at many sites. So there's perhaps 14 sites in the Oslo area that we think might have had populations established due to this stocking. And we're currently undertaking a genetics project to actually determine whether that's the case. And it's probably the case for some of these sites, but maybe not for all of them. It's not in a restoration effort, but it does show that stocking of infected fish can lead to the establishment of mussel populations and therefore can be used as a restoration tool. Now, the Hammerbekken stream is a small stream in central Norway, just outside of Trondheim, the biggest city up there. And data from 2008 to 2010 show there's less than 30 live eastern pearl shells in the stream, and there's no recruitment. And in the map, you can see the blue lines at the top of the map, and at the bottom of the map, they indicate the two distribution areas within the stream. Infections were done in 2008 to 2010 at a fish breeding facility. After the infections, they released almost 4,000 juvenile trout into the stream. The subsequent year, they managed to recapture on average 70% of the infected trout. And based on these data, the infection intensity on the trout, they were estimating that 90,000 juvenile mussels were actually released into this stream. To evaluate whether this actually led to recruitment in the stream, in 2020, 10 areas that you see in the map to the right, the blue dots, were excavated. And the reason they were excavated is that, as I mentioned, juvenile mussels live in the gravel. So if you want to find them, you got to dig. And at those 10 sites, about 30 mussels were found. In addition, 10 more recruits were found between the excavation areas. And this really illustrates sort of the effort you have to go to be able to use this method to restore mussels to a river, or a stream in this case. So 90,000 mussels, juvenile mussels, were probably released into the stream, and that's resulted in 41 mussels. Now, of course, that's an underestimate because it's only smaller areas of the streams that have been excavated and surveyed, but probably there's only a couple hundred, maybe several hundred, maybe 1,000 mussels that have established and survived in the stream from those 90,000. And here you see a photo of some of those mussels that they found. I'll move on to the cultivation and stocking of juvenile mussels. This is a big restoration effort undergoing in Norway. The cultivation part of the method, it's run by the University of Bergen, by Per Jacobsen. And we basically used the Czech method, sort of a further development of the Czech method. Our experts sort of were trained by Mikael Lange from Germany, who is also sort of developing his work based upon the Czech method. And what we do is we collect adult mussels, we infect juvenile salmon and trout, and in the top photo you see tanks that the infected fish are kept in, and once the mussel juvenile mussels drop from the fish, they're collected in sieves below. After that, they're maintained in boxes for 5 to 6 weeks. You see one of these boxes in the middle, and they're fed with algae and chironomids. And then they stay for 2 years. That's what we've ended up doing in these channels that you see in the bottom photo. Now, the stocking and monitoring is run by Nina, my institute, and by a company called Consulting Biologists. We've tried stocking anything from 1 to 7-year-old mussels, and you'll have to apologize for my lack of a Czech pronunciation. We use Ruska boxes, as we call them, to do the stocking, and you see them in this photo. They're flow-through boxes that are filled with gravel and mussels. And here you see them in the river. We use metal poles that we drive into the river bottom, and then we attach the boxes to those poles so they stay put at our stocking sites. And we do this stocking in spring and summer, and then we monitor the boxes after 6 and 12 months. And when we do the monitoring, we look at survival and growth of the mussels, and we do that by taking photos of the mussels in a petri dish with millimeter paper background. Allows us to count the mussels and measure them in the lab afterwards. And then after the 12-month monitoring, we release the mussels freely into the river or stream. Now, we've stocked at 18 sites in central and western Norway, as you see in the map. We've undertaken 23 separate stocking efforts, and more efforts are underway as we have more populations being produced in the cultivation facility. Now, I'll give you one example of our stocking efforts. Data from 2008 and 2015 show that there were approximately 3,600 live eastern pearl shells in the Slørdalselva River, and that there is very low recruitment. And in the map, you can see it's just a small river between 2 lakes. The mussels were found between the upper lake and Station 5. Now, we did 2 separate stockings. We stocked in both 2017 and 2018. In 2017, we stocked 300 mussels. Half of them were produced on salmon and half of them were produced on trout. And the reason we did that is we didn't really know whether this was a salmon mussel or a trout mussel. So we tried to infect both in the lab, and we got heavy infections on salmon and low infections on trout, which is very typical if you try to infect salmon and trout with a salmon mussel. Then in 2018, we released 666 2-year-old mussels, and these were all produced on salmon. And this figure shows you the survival of the stocked mussels in the river. And the bottom line is the 1-year-old Atlantic salmon mussels. The middle line from brown trout mussels, and the upper line is the 2-year-old Atlantic salmon mussels. And all these differences in survival were significant. Although on a whole, they're actually quite a bit lower than we typically find when we stock 2-year-old or older mussels. Then we typically have had survival in the 80s or 90s, rather than here where 2-year-old mussels had a survival at about 50%. So why did we get this difference in survival? Well, we think it has to do with the size of the mussels when we stocked them. The 1-year-old mussels that were produced on salmon were significantly smaller than the 1-year-old mussels that were produced in brown trout, which were significantly smaller than the 2-year-old mussels produced on salmon. And we think that maybe the reason why the mussels produced on the trout were bigger than the mussels produced on the salmon is that there was a lot less larvae attaching to the trout, so there's maybe less competition between the mussel larvae for resources on the trout than on the salmon. So to give you sort of a summary of the lessons that we've learned from our stocking efforts, is that generally we've had good survival and growth. As I mentioned, typically we've had over 80% survival for the year following stocking. We've also found that juvenile mussels should be at least 4 millimeters long and 2 years old when you stock them. If you stock mussels smaller than that, we've had bad results. And we've also found that you should release the mussels in summer rather than spring. But that might not be as applicable in the Czech Republic as in cold Norway. So to bring you to the conclusions of this talk, we've had positive results of stocking host fish, especially where we've had salmon mussels where the salmon had been lost. When we've done infection of fish with mussel larvae, it doesn't seem to have been that successful when we did these in-river infections, while actually doing infections at breeding facilities for fish where you can produce a lot higher numbers can have a positive effect. However, you need to produce a lot of fish infected with mussels, and the conditions in the river have to be good otherwise for this to work. Now, the removal of migration barriers, again, has been positive, especially when it's allowed salmon to return to its natural areas, giving salmon mussels their hosts back. Relocations of mussels, well, we've had some cases where if you relocate a mussel from one river to another river, we've had one case where the mussel established and started reproducing, but in most cases they've either died or there has been no recruitment. Biotope improvement, we don't really have the data to evaluate that. The same for livestock impact mitigation and for buffer zones. Now, the cultivation of stocking of juvenile mussels can definitely work. We've had some issues in the cultivation part, but they've been fixed now, and it seems like once we actually manage to produce juvenile mussels, they survive in the rivers. However, we still need to follow up with long-term studies to look at whether that survival in our boxes continues once the mussels are released into the river itself. When it comes to hydropower and environmental design, we've had some positive effects of increasing minimum water levels from hydropower plants, but further studies on how to do environmental design for hydropower plants is needed. And we've also had some success of liming acidified rivers, leading to the recovery of mussel populations in those rivers. So I'll leave you with Nina's slogan, cooperation and expertise for a sustainable future. I would like to thank Per Jacobsen, who runs the cultivation facility at the University of Bergen, Steinar Kolos and Ingrid Vatna, who are my collaborators on the stocking program, Bjørn Meid Larsen at Nina, who's the grand old man of freshwater pearl mussels in Norway, and who also undertook the stocking of the infected fish in Hammerbekken. And then I would also like to thank Kjell Sandås at Natural Science Consulting, and Jørn Enerud at Environmental and Fish Services. They've done a lot of their relocation efforts within rivers. And I would also like to thank you for listening to this video presentation on the restoration of freshwater pearl in Norway. I hope that you've learned something, and then maybe some of our lessons can be useful to the restoration of the freshwater pearl mussel in the Czech Republic. Thank you.